everybody. I am Alexa Tinsley. I'm the Director of Programs here at Jett Foundation. I want to thank you all again for joining our community series, webinar series today, as we welcome Solid Biosciences to discuss INSPIRE Duchenne and their clinical trial for SGT-003, which is a next-generation microdystrophin gene therapy for individuals living with Duchenne muscular dystrophy. We are absolutely 100% grateful to our partners like Solid for helping us make these webinars possible and bringing the information right into your homes virtually. Unfortunately, Annie is not able to be on with us this afternoon, but we do have Patrick Gonzalez, Senior Director of Clinical Science at Solid, and then Matthew Harmlink, who is the Medical Director and Therapeutic Head for the Neuromuscular Program. So before we get started, I do just want to mention a few things. This webinar is being recorded, so if you have to log off for any reason, if there's anyone that you know that wasn't able to attend, this webinar will be recorded and it will be on our YouTube that you can watch later. We typically do stream to Facebook Live, but are not able to do so today, but we will have it on Facebook for your viewing later, so you can watch it on YouTube or Facebook after. And you can also share it through those two platforms as well. At the end of the webinar, we will have a Q&A. There is a question box in your panel. For me, it's at the bottom right. It looks like a little chat box with a question mark in it. So we do ask that if you have any questions throughout the webinar, please feel free to drop them into the questions box, and we'll do our best to answer your question. We do our best to get through all of them, but if for some reason we aren't able to, we will record them and we will send them to Solid to see if we can get answers to your questions later on. If for some reason your question does go through the chat, we do also try to get to those, but we do primarily read them through the Q&A box. So please, if you can, try to put them in there. There is also closed captioning and language translation. If that is something that you need, you can enable this by clicking the CC button and enabling transcription in your preferred language. I believe there's an auto language button that you can actually toggle, and then it has a list of languages that you can pick from. So that's a lot of talking from me, but I will go ahead and hand it off to Pat.
Perfect. Thank you, Alexa, and thank you everyone for joining today to go through this update. We're very excited to be here and to be able to provide this additional information to all of you, and then also take questions at the end of the presentation. So as Alexa mentioned, this is our group who is here. We're sad that Annie wasn't able to join us today, but we'll do our best to be able to fill in for her. As we get started, we are a public company, so just always have to include the forward-looking statements slide, that we will be making some forward-looking statements throughout the presentation. And so I really just want to introduce the company first to those of you who may not know Solid Biosciences as well, and then get through an overview of really what is our focus today, our SGT-003 program that we are developing for the treatment of Duchenne. I'll go through the background and really the data that we generated to date on the non-clinical side as well with research, and then I'll hand it over to my colleague, Matt Harmelink, to be able to go through our trial that's currently ongoing and the updates that we're providing today. So at a whole, Solid Biosciences is a life sciences company that is developing precision genetic medicines for the treatment of neuromuscular and cardiac diseases. So for those of you who don't know us, we were founded in 2013 by those who were directly impacted by Duchenne. So Annie Gano, who is unable to join us today, was one of our co-founders, and really a lot of this stemmed from their diagnosis of their son, Eitani, with Duchenne muscular dystrophy. And so with that, we've really grown from the initial point now with a team that has been building towards additional neuromuscular indications as well as cardiac indications. And so with our platform focus really on gene therapies that could be disease modifying. With that, we've really looked to be able to grow our initial base from that focus on Duchenne and really trying to impact as many different rare disease indications as possible. And because of our core of how we started, we've always wanted to maintain really strong relationships with patient advocacy organizations and the community. And so this is why we're always so happy to be able to have these sorts of opportunities to talk directly with the community like this. Our company is headquartered out of Boston, Massachusetts, and a couple of us are especially located remotely. You'll always see us at meetings throughout the year as well. So we're always very fortunate to be able to see and talk to the community as well. So going directly into the focus of today's talk is SGT-003. And as Alexa mentioned, we call this a next generation microdystrophin gene therapy for Duchenne. So we'll go through a little bit of the background on kind of what initially is that gene therapy, what are the core pieces of the SGT-003 approach that we consider this a next generation gene therapy, and then what are the data that have really made us excited about moving this into the clinic and now quickly expanding into additional sites as well. So overall, just at a high level, a gene therapy is really a way to be able to restore functionality. And through the approach that we're taking is using a capsid, which is really a delivery vehicle, to then drive the expression of something that we want to express in a tissue, here being a transgene. And so different approaches will have different things that are being delivered. Because we are focusing on Duchenne muscular dystrophy, we are looking to replace the protein that's absent in Duchenne, which is dystrophin. For our specific approach, and this is our SGT-003, we have a couple of core pieces really of what makes up SGT-003 that we want to highlight, as well as also an important piece of this in really making the overall product that all have additive differences that make it a next generation approach. So starting from the left here is really what is being delivered through this. And so I think all of us know that dystrophin is a critical membrane protein that is really the reason for why Duchenne muscular dystrophy occurs is the loss of this. And so because of its size, when we're using an approach like an AAV-based gene therapy, we need to be able to choose pieces of that full-length dystrophin, which is really known as the largest protein encoding gene in the human genome, to then be able to fit within this delivery vehicle. And so different choices that we make are really based on foundational studies that have been done through experts in the field, those from Kay Davies and work that was done with Becker muscular dystrophy, Jeff Chamberlain in really identifying initial microdystrophins. And unique to our construct here is Dongsheng Dewan's input also in looking at what domains have different functionality, and that being the NNOS binding domain that's unique to the microdystrophin in SGT-003. And so with this, we know that we have certain structural domains that we have to maintain, but this inclusion, and we'll go through it a little bit more on the next slide, is really hoping to be able to have that additional function that could also be important for this type of approach. And with this, as we're using an AAV-based gene therapy, each AAV has a different propensity for getting to different tissue types. And so when we're looking at these different capsids, we call each of these different versions serotypes. And that really means that there are naturally occurring serotypes, there are those that have been rationally designed, ones that can potentially get to different levels in different tissues. With the version that we're using here, which we call AAV SLB101, this is a novel muscle tropic capsid. And what does muscle tropic means? It actually means that we're getting more of this when we have a systemic delivery of our gene therapy, we get more of that to muscle tissue. So as we looked at what first generation approaches were able to achieve, we did know that there was a good amount that could get to muscle, but with the high systemic doses that we're administering, we know that we could do better, and we know that being able to reduce a dose could also be important for safety. And that really pushed us towards wanting to use a next generation capsid for those reasons. And what we saw from the studies that we were performing was that we were actually able to see multiple fold improvements in the expression of this transgene of our microdystrophin, here showing a non-human primate study in our target tissues, as well as reduced levels getting to the liver. So hopefully through this approach, aiming to be able to improve on both the effectiveness of this therapy, as well as potentially on the safety of it as well. And then with this, when we put together this SGT-003 and really any AAV gene therapy, what we have to have is a manufacturing process that's able to make enough to be able to get for each of these large systemic doses that we're administering, and importantly, to be able to treat all of the patients that we can. And so this has been a major focus for Solid from the very early days. And what we wanted to do with this program was really optimize the manufacturing process to a point where we would be able to reduce the empty particles that were part of this. because as you make an AAV, there are both full particles, these are the ones that have what you're trying to deliver, and then also empty particles that are part of the process, and that can be at different proportions based on the level of purification that you may do and the process in general. So one of our big focuses was wanting to reduce those empty particles so that what we're delivering is really focused on those full particles that can express microdystrophin. And what we've seen from early studies were that as we increase that amount of full particles, we know that we're overall going to reduce the total viral load, so the total amount of particles that get to throughout a body. But we also see that will also additively improve on the effectiveness of this. And so putting all of these pieces together for SGT-003, we're really hopeful that these additive benefits could then create a meaningful differentiated product for Duchenne. So we talked about the microdystrophin a little bit, but to take a quick focus on really what is that full-length dystrophin and how does a microdystrophin, and specifically the one used for SGT-003, compare to that? So I mentioned that there are structural pieces of a dystrophin, and here you can see on the top that it's a very, very large protein that has lots of different domains. The two that are really thought of mainly are going to be that intracellular portion, so the part that links the inside of a cell, and then this part over here that's called the beta dystroglycan binding domain, which really links to the membrane. So when you have these two pieces in a dystrophin, you're able to form that structural stability that you need for a muscle to be able to withstand the contraction and relaxation that occurs on a day-to-day basis. When this is absent, you're then going to have that susceptibility of a muscle to actually degrade over time because it's not able to be able to withstand that process. So what we want to do with the therapeutic approach is to restore that core functionality. With that, we also know that there's a lot of other functions that are within the protein itself. And so you can see here in blue, all of this is considered the rod domain of dystrophin. And so many of these are what we call redundant functions. So some of them can replace the function of others. However, some of them are also unique in function. And so one of these items that's critically unique is the R16, R17 piece of this rod domain. And it's unique in that it's able to actually restore nNOS localization to the membrane. And so why does that matter? It's that nNOS itself is a signaling molecule. And so when nNOS is present in a location, especially towards the outside of the cell, so at the membrane, it's able to then have a result of having nitric oxide production when it is activated. And so in situations where you might have higher energy demand for a muscle, so if you're exercising, if you're moving, and you need more blood flow to get to the muscle, this is where nNOS activation is necessary. Otherwise, you're going to have a lack of oxygen to that muscle, which could result in injury and damage. And so the ability to include this domain within a microdystrophin has really been identified through a lot of early work from the non-clinical side and also looking at Becker muscular dystrophy as potentially important for being able to result in that added function of the full-length protein. And really that's been a core piece of why we've included this in our microdystrophin for both structural function and then signaling function through that extramolecular domain here. I mentioned what SLB101 is, this novel serotype that we developed, but really to go through the process here, what happened was we understood that there are these naturally occurring serotypes like an AAV9, like an AAV8 or RH74 that have different ability to get to muscle. And what we wanted to do was start with a basis of AAV9, look at ways that we could be able to modify this capsid to get to muscle better. And so what you can see here is that through different iterations that we were able to evaluate, first in the early models in cells in dishes, then into some of these early small animal model studies, we can see that in choosing this capsid, the AAV-SLB101, this is really because we're able to observe a profile where we had multiple times higher levels getting to the muscle. Here looking at quadriceps and also the heart as representative skeletal and cardiac muscle here. So key target tissues for Duchenne treatment. We have these multiple fold higher levels, and this is something that could be potentially optimal and beneficial for a treatment for Duchenne. And then at the same time, what we saw was that as we are getting more to the muscle, so as we're actually targeting our muscle better, we also had less getting to off-target tissues, so like the liver. And we know that when we have a large systemic dose of a gene therapy, a lot of this does go to the liver because that's really how your body's clearing out and where there's really a sink for this to be able to accumulate. And so if we have the ability to lessen that amount that gets to the liver with these doses, we could potentially improve on the safety side. And so overall, this profile is really what we wanted to look at for a next generation capsid approach and put into our program. From these initial non-clinical studies that were done in that mouse model of Duchenne, we then wanted to see how does this scale up when we look at something that could be closer to a human. And so we did this in the non-human primate, where we looked similarly to the other studies at single intravenous administrations of a capsid with SLB101, and here looking at a reporter transgene which was used. And what we could see is that compared to AAV9, we saw a very similar profile as what we saw in those mouse studies, where we have multiple fold higher levels that are getting to the skeletal muscle as well as cardiac muscle for SLB101 compared to AAV9. At the same time, we were also able to observe very excitedly that same profile where while we get more to the muscle, which is a good thing, we also get less to the liver, which is also a good thing. So both of those items that we initially characterized were able to scale up into the non-human primate. And what we wanted to do from there was also compare against really what else is there that we know is going to be something to make a decision against and to really see how does this treatment compare to available options in the field. And so what we did was packaged our microdystrophin within SLB101 and also within an RH74. So as I mentioned, one of the first generation naturally occurring capsids. And what we were able to observe here was similarly to AAV9, we also saw that AAV-SLB101 demonstrated multiple fold higher levels of expression. And here, this is specifically looking at microdystrophin expression in heart tissue and quadriceps tissue. So representative target tissues that we would want to see this profile in for a treatment for Duchenne. And so here looking at about 4 to 6 times higher levels of expression compared to that RH74, really this was one of those important studies for us that would laid the foundation for moving this program quickly and wanting to get this moving towards the clinic as fast as we could. And so with that, we then moved into our IND-enabling non-clinical studies. So what was required to be able to characterize both the safety and the efficacy from a non-clinical side to then be able to enable the progression into clinical studies. And as I mentioned, what we wanted to see really was the initial functionality of a microdystrophin for the ability of these different domains that it has to actually show that they're doing what they're supposed to be doing. And so we look at immunofluorescence here, so using an antibody to identify the microdystrophin protein. And what we can see in these sections from muscle tissues are that as we increase the dose of our SGT-003, we also have increasingly higher levels of microdystrophin expression, and it's doing what it's supposed to be doing in that it's localizing to the muscle membrane. So with those different domains that are present, the intracellular part and the extracellular part, this is able to actually localize correctly. And because of that unique domain that we talked about, which was this R16, R17 nNOS binding domain, we also importantly look at nNOS activity. And so here, using a different histology stain on a muscle, what we can see is that as we have higher levels of microdystrophin expression, we also see that those fibers with that microdystrophin expression are now being able to have nNOS enzymatically active at the muscle membrane. And so this is something that increases in a dose-responsive manner. And you can see that in this MDX vehicle here, so this dystrophin negative model of Duchenne, whereas this is really negative at the muscle membrane for nNOS as well as microdystrophin, we have those increases that correspond with the dose over time. And so we looked at that from a visual sense, but we always want to quantify it as well. And when we think about a muscle, we know that the entire muscle, if we can really get to the majority of it as much as possible, we can have a more pronounced effect in that we can protect more muscle. And so when we look at the different quantifications at different time points from these studies, what we saw is that actually very early on, and here this is day 4 in a study after dosing, that we did already begin to see quite robust levels of microdystrophin expression that occurred in a dose-responsive manner. Interestingly, by a month out, what we can see is that we really have about 100% of positive fibers. So the entire muscle is showing microdystrophin expression at all of these dose levels that we evaluated here. So in this study, 3E13 as the lowest dose, up to 3E14. And what we actually identified was that it took even lower levels than this to start to see at these later time points less than 100% positive fibers. So this is very encouraging for us. And you can see that this is the profile throughout all of these muscles that we evaluated here. The quadriceps, so upper leg, your gastrocnemius, which is your lower leg, diaphragm, and then heart, as we know, are all important muscles for different functions for Duchenne. So looking at that molecular functionality, we know that microdystrophin is localizing where it's supposed to, it's recruiting the proteins back that it's supposed to, and it's doing that in a dose-responsive manner. What we wanted to see is now from a physiology sense, what can we tell if that microdystrophin is also resulting in a functional difference for these muscles overall? And so we look at this in a couple of different ways in these studies. The one on the left here is looking at treadmill exhaustion, which is really an endurance assay. So it's saying, are you able to function continuously over time? And what we can see is that we had a significant improvement, even at that lowest dose level tested at that 3E13 over untreated vehicle treated animals. And then importantly here on the right side, looking at the actual force that a muscle can generate, what we can see is that we have a similar profile where all of the dose levels tested are significantly improved, and this is occurring as low as that 3E13 lowest dose tested. And this is important to highlight as well, because if you can see this level here, it really is restoring that function, the force of the muscle that the muscle can produce back to a wild type level. And so this is, again, a really exciting result showing the potential meaningful difference that we could have from a treatment with an SGT-003 from these non-clinical studies. And then when we think about the impact that we could have overall for a treatment, we also want to look at biomarkers. So we looked at microdystrophin expression before, but what else can we look at from a circulating sense? So if we were to take a blood sample, what can we look at throughout the way? One thing that I think all of us really understand well is serum CK, so creatine kinase, usually used as a diagnostic for Duchenne, and something that we know is going to be really high in our patients over time. And what we can see here is that following treatment, we have a significant reduction of CK, very near these wild type levels as well. So a meaningful difference here from a biomarker that we understand well for for Duchenne overall. In addition, we're starting to look at other exploratory biomarkers and looking to see how they may correlate with these markers that we know a little bit better to be able to potentially put together a supportive way of evaluating the overall functionality of the protein. And so what we can see here in looking at serum titin, so this is a marker again that we're looking at in circulation, we can see that similar to CK, we have that significant decrease in serum titin as low as this lowest dose level tested, and it really shows that similar profile right down to levels that are near wild type as well. In addition, we look at AST. So a lot of the times when we look at these different markers in Duchenne, we have to consider this differently than others because AST, ALT, some of these markers are going to be elevated, and a lot of the times they're considered liver function enzymes. But here in Duchenne, we know that they're leaking rather than a result of the liver. And so what we can see is that when we have this membrane stabilization that we have from the microdystrophin expression, we're also seeing that we have a reduction in this biomarker as well. So really from the functional side as well as from a molecular side, seeing a really exciting profile overall for this treatment. And so we were happy to, at the end of last year, announce that we had clearance from the FDA to move forward with this program into clinical trials. We do believe that this capsid, AAV-SLB101, represents one of these first rationally designed capsids, and that the program overall is a next generation therapy because of these different components that I just went through. We see that from a functional side and from a biomarker side, we have these improvements overall, and we're really excited to be able to move this into the clinic with our study that initiated this year. And so I'll hand it over to my colleague Matt to be able to go through what we are calling the INSPIRE Duchenne study, which is active enrolling currently.
Thank you, Pat, and excited to talk more about the trial that's ongoing. So if we can go to the next slide. So INSPIRE Duchenne is a phase 1/2 clinical trial to evaluate SGT-003. It's going to be conducted in the US, Canada, and Europe with a single dose of 1E14 viral genomes per kilogram in an open label study, meaning there's not a placebo arm. Everyone who's in the study is dosed. It's a study for ambulant patients with Duchenne's muscular dystrophy, and the prophylactic immunomodulation is increasing the daily steroids to a little bit higher dose around the time of dosing shortly after that. The key analyses in this trial is safety at day 360, looking at microdystrophin expression at day 90, and the time to rise functional assessment at day 540. We've divided the study into two cohorts. So cohort 1 is boys who are age 4 to under 7, and cohort 2 is for boys who are age 7 to under 12 years of age. Both cohorts are actively enrolling currently. You do also have to be at or under 50 kilograms of weight, and the goal total enrollment is 43 patients. Next slide. So as I mentioned before, we do have these two cohorts. Now we do have some DMD mutation exclusions. So we are excluding any boy who has a deletion between exons 1 to 11 or 42 to 45 inclusive, including if the deletion extends beyond that. So for example, if you have a 43 to 47 deletion, you would be unfortunately excluded from the trial. For functional assessments, there's no functional inclusion or exclusion criteria you have to meet for cohort 1, but for cohort 2, there is a time to rise and a 10-meter walk-run criteria. Anybody coming into the study has to be negative for AAV antibodies, and you cannot have priorly received a gene therapy treatment, whether it's investigational or commercial. There is a washout period for exon skipping therapies, vamorolone and or givinostat of at least 12 weeks prior to entering the study, but you cannot be on those during the treatment period of the study. For steroids, we're including boys who've been on a steroid stable regime of daily oral steroids of prednisone or deflazacort for 12 or more weeks prior to entering the study as well. If you are on steroids that are different than that before 12 weeks, you can be adjusted to that daily steroids for at least 12 weeks before you come in. Next slide. Because this is a phase 1/2 trial, one of our main goals is getting robust safety and then some preliminary efficacy analysis. For safety, the first 30 days, everyone will have frequent visits to the hospital or their clinic to get checkups and blood work to monitor for safety. Afterwards, these visits gradually become less frequent, but will continue for 5 years post-administration as required by regulators. For an efficacy measurement, there are 3 total muscle biopsies to be performed during the study, with ideally a needle biopsy technique at screening, day 90, and day 360. We're using this for evaluation of microdystrophin expression. Testing is also being done for motor functioning, pulmonary functioning, and other patient-reported outcomes, so not just the time to rise. These are done by in-clinic questionnaires and testing, and there will be also an at-home video and activity monitoring portion as well. Next slide. So we have already, because this study is enrolling, we have already achieved some milestones. So we had our first clinical site activated in Q2 of 2024. Our first participant was dosed with SG003 in Q2 of 2024 as well. And we've had 3 participants dosed by the end of Q3 2024. To date, there have been no serious adverse events reported for the participants who received SGT003. Of note, if you do follow the study, you will note that there was an expansion from a smaller safety study previously in the year into a larger study targeting efficacy as well in Q3 of 2024. Now, upcoming activities, we currently have active sites that are screening and enrolling patients, and upcoming active sites are identifying patients for potential participation. We are looking to have summary 90-day data for the first 3 participants to be shared in Q1 of 2025. Next slide. As I mentioned before, also, we are looking at clinical site expansion that's underway. In the US, there's multiple additional sites in study startup with expected new activations starting this quarter and continuing onward. We do have 1 site in startup right now in Canada. The clinical trial application was cleared by Health Canada, and we're expecting that site startup any day now this quarter, this year. Additionally, we are looking at sites in Europe that are in parallel with the regulatory activities. So clinical trial applications are submitted with expected site activations in the first half of next year. New slide. Here you can see again the current sites that we have. Looking across the country, we have sites everywhere from, for example, California, Ohio, looking at Virginia coming on very shortly, as well as Canada and Arkansas, and more sites to come in the future. Next slide. And if you're interested in hearing more about the study or potentially being a participant, feel free to reach out to the sites. This is the contact information for the sites that are either already active or being activated very shortly. Additionally, if you have information that you want to know about the study, you can use this QR code. This sends you to the clinicaltrials.gov link, which is being kept updated throughout. In addition, we also have our clinicaltrials@solidbio.com email for questions. So with that, I'm going to open up for the Q&A and any questions.
Thank you guys. Really, truly appreciate you, bringing this to the community. I think there's, so much going on, but to know, what's different and what you guys are doing is really important. So truly appreciate you sharing with the community. We do have one question. I'm curious about why there's a washout period for vamorolone, but not prednisone or deflazacort, being that it is also a daily steroid. Can you elaborate more on the reasoning, which I think is also an interesting point as well?
Yeah. So there was a lot of discussion about vamorolone, about the data that's out currently and its usage. Our feeling at this time is that we wanted to make sure that we have a full comprehensive view of the steroids that are being used. Deflazacort and prednisone has been used for a long time as well, whereas vamorolone is a little bit of a newer steroid. And so based off, again, being very cautious about the safety perspective as well as efficacy, we wanted to know that. Also, there's more long-term data on the other two steroids. So that was sort of the determination. We talked with a lot of different experts across the field and sort of to obtain all this information to go forward.
Perfect.
Yeah. And one thing I'll highlight too is that while it is a washout of vamorolone, what we want to do within the study is also make sure that there is flexibility for participants. So the first 18 months, which we're really considering that important functional endpoint, that's where we would want this to be maintained, this regimen to be maintained of deflazacort or prednisone. However, after that, we do allow for there to be flexibility for some of these other things that patients may want to be also taking or switch to.
So here's a question from my seat that you may or may not be able to answer. But so say someone is on vamorolone but they have to be on daily steroids or a solid dose of steroids and steady dose of steroids. Obviously go to your doctor and have that discussion, but they would have to transfer to another one is what I'm assuming. Yes.
That is correct. Yes.
Just to be clear.
Yes, as Pat mentioned, the treatment period. If we think of this as the 5-year study, though, outside that treatment period afterwards, we are going to be more flexible for patients. We also know 5 years is a long time and the field is changing very rapidly. So we don't want to be exclusionary for that entire period.
Absolutely. Thank you, guys. Question, what about early deletions like exons 8 or 9?
Another good question. Currently, those early exons are excluded. 1 through 11 is excluded. It's not that we're not looking at that very carefully, but currently, as this is our first in human safety study, we want to be overly abundant cautious as we move forward. As we gather more data, that's something that will be discussed in the future.
Awesome. And then how will immunosuppressant regimen be pre and post dosing?
So the immunosuppressant regimen is increased prednisone. So that starts around the peridosing period and then continues and is weaned off over time back to the baseline steroids. We don't have other medications currently in the trial as preventative.
Perfect. Awesome. Any other questions from anybody? I'll give it another minute to see if anybody has any questions. But again, thank you both. Super interesting. So thank you guys for sharing.
Maybe as we're holding on this slide, I'll also kind of emphasize just we went through a lot of that non-clinical data. And one of the important pieces for us was wanting to look at what's really the dose level that we'll be using in the study. I think a lot of what we've seen has shown that we saw efficacy in these models at much lower dose levels, actually. But we know that currently everything doesn't always scale up 1-to-1, and there's a lot of room for improvement, I think, overall. And so when we chose this dose level, what we wanted to do was target something that could be potentially optimally efficacious rather than looking at something that could have a minimal effectiveness. And so really that's the reason for this, reducing the dose level overall, but also trying to have a meaningful difference.
Awesome. And then a question that we had beforehand that I mentioned previously, but I do think that I would like just to have out there in terms of, is this available for women, which we know is no at this point in time, but is it something that is being discussed in the future possibly?
So treating women with dystrophinopathy mutations and variants is a bigger discussion, I think. And certainly with this first in human study, we're looking at this very selected cohort of patients. That I think is going to be a bigger discussion that has to happen, as well as some natural history development, things like that, before we can move into that area.
Absolutely. Well, thank you guys again. If anybody does have any questions, I know sometimes it takes some time to process, and then, you know, like I said, it will be available after the fact as well. So if you do feel you need to rewatch and then have questions after that, please feel free to reach out to clinicaltrials@solidbio.com, or you can reach out to me, Alexa@jettfoundation.org, and we will definitely see if we can get those questions answered. Again, I appreciate everyone so much for being here today. I hope you will join us for our next one, and we will see you all soon. And thank you, Pat and Matt, for being here and helping educate the community today. I appreciate you both.
Thank you for having us.
Thanks so much.
Have a great day,
Everyone.
everyone.